Hi, this is Mrs. Whalen. It's October 10th, 2016, and I'm doing a quarter one review for all of my biology classes, Biology 1 and Biology Honors, for all of quarter one, since I haven't been there the last couple of weeks. So I'm going to spend just a little bit of time going over some of the things that I think you might need some extra help with. Don't forget that there's the SWAY presentation that I introduced during class today through the sub, and the review sheet that you would have gotten during class today or that you'll be receiving tomorrow, Tuesday. Your quarter one test will be on Wednesday and Thursday. Hopefully I'll be there to see you. If not, no worries. That's the day that everything is due for ecology. That's Wednesday and Thursday, along with your Unit 1 test that you're going to be taking and the benchmark test from the county. Just remember that when I'm doing my grades on Friday for record day, when you have the day off, I will take into consideration the fact that I haven't been there the last couple of weeks, and I will be gracious to you as long as you can be patient and gracious to me while I am healing and recovering from being sick. Hoping to be back with you guys as quickly as possible. So, I guess we'll start from the beginning, which was scientific method. I think most of you, based on what I've already seen during the course of quarter one, are in good shape as far as the scientific method is concerned and the vocabulary that is associated with it. Just make sure that you go back and review those things that you are already tested on. For example, remember how we talked about if we wanted to find out how do different types of soil, for example, affect plant growth. We said that was a possible question that somebody could test scientifically. And remember, you should be able to, A, make a hypothesis for that experiment, okay, and know what the whole steps are of the scientific method, from I asked a question, do different types of soil affect plant growth, to I need to make a hypothesis, my educated guess, which means there's an educated guess, so I have to do some background information research before I do that. After you've made that hypothesis, a good scientific hypothesis should be testable somehow. This one is. We could say, I think that different types of soil will have an effect on plant growth. And you could even go so far as to say which soil type you think might be best based on your research. And then, as you're setting up the experiment, you should be able to identify what's the dependent variable, independent variable. So for example, you are measuring the growth of that plant. So you would say maybe every day check the length or the height of that plant, and you could record it in centimeters. So that plant growth would be your dependent variable. Remember, that's the outcome that you're measuring from the experiment. And because it's a number, like 2.1 centimeters, for example, that would be quantitative with an N kind of data that you're collecting. Now, if I said something like the leaves turned yellow, or the plant shriveled up, or the plant died, those kind of adjectives, descriptive words, remember, are going to be qualitative with an L kind of data. Meanwhile, so you're doing this experiment, you're collecting your data. We talked about the dependent variable. Remember that the independent variable is that one thing that you as the experimenter are going to test or change. Remember, there can only be one in a good experiment. And so for us, that one thing that we're testing or that we're changing is your soil type. Sorry if the writing's a little messy here. You're collecting data, probably recording it in a data table, nice and organized. And then when the experiment's over, you get into the analysis part of it. So you're looking at the data, maybe even creating some graphs. Remember that when you set up a graph, then on the y-axis, going up and down, should be that dependent variable, like from this case, your plant growth. But on the x-axis should be whatever the independent variable is, like your soil type, etc. You plot your points, you draw your line, then you have a nice picture of the data so that you can interpret the results easily and communicate it to other people. So now this communicate to other people part, now that you've looked at your data, you're starting to form a conclusion, okay, I either accepted or rejected my hypothesis. And then you want to communicate it to others. Remember that step of peer review. Who are your peers? Anybody who can review your work. Hopefully somebody that's from a similar background so that they know what you're talking about. And they're going to see, hey, maybe you made an error that you didn't think about, like we had discussed in class previously. After that, if people do these experiments over and over and over again and they keep getting similar results, remember that over time we could create scientific theories, which is not the same thing as me saying, oh, I have a theory about why I've been sick for so long. That's different than a scientific theory. So we've done lots of experiments, we keep getting the same results, somebody else can repeat the experiment and they get the same results, and so we say, hey, that's good science, and we're pretty sure that we can make a theory now about this. Just remember that theories are not facts, they're not absolute truths. They can change over time, just like we said that there was a theory that the world was flat once upon a time. That was a great scientific theory at that time, as we discussed before. But new evidence came to light, just like happens constantly in our world, new technology, new experiments, new data, and we say, oh, hey, people came back from that voyage. I guess they didn't drop off the edge of that flat line like we had talked about before. So then they have to come up with a new theory. And that's kind of your scientific method stuff in a nutshell. See your review guide and SWAY and your class notes and assignments for more details if necessary. Don't forget that throughout all of this and throughout this week, as always, you can Edmodo message me, email me, and I will get back to you as soon as is possible. Okay, we talked about microscopy. And remember, there's two basic types of microscopes. Light microscope and electron microscope. The light microscope is great to view most things, okay? We can magnify them and see lots of great detail. However, if we want to see something that we need a lot more detail, we need to magnify it maybe 20,000 or 100,000 times, then that's when we want to use one of those electron microscopes. And with those electron microscopes, remember there's two of them, the TEM and the SEM. Remember the transmission and scanning electron microscopes. The upside to the electron microscopes is you get great detail. The downside, remember, in the processing of the samples, it kills the living thing if you wanted to view something that was alive. So you can view organisms, you just can't view them when they're actively alive doing all their life processes. So later on in the year when we look at pond water samples under the microscope and we want to see the organisms swimming around and how they're eating and how they're behaving, we'd rather use the light microscope because they're alive and we can watch them doing their life stuff. Besides the fact that we don't have an electron microscope because it costs as much as most people's house, right? We talked about that before as well. Okay. Remember that you calculate your total power of magnification by multiplying eyepiece times objective lens. Real nice and easy when you're using that light microscope. And that, remember, there's a diaphragm, that little part on the bottom of the stage of the microscope, and you can turn that and use it to adjust the lighting to help you see things better, give you a little bit better contrast. Now, we also discussed part of that first test, characteristics of life. How does a scientist know if something is actually alive? And we decided that there has to be a good way to do it. So that, let's just say we find out that there's something new we've never seen before. And we don't know. Is it alive? Is it dead, meaning it was alive at one point, it's not anymore? Or is it inanimate? Is it something that was never alive to begin with? So remember, a biologist knows if something's alive by looking at, we came up with a list of 8. Remember, we said the number doesn't matter, the characteristics do. We came up with this list of characteristics something has to have in order to be considered alive, or was alive at some point. And we said, has to be made of cells, highly organized, they have a genetic code, like that DNA, RNA, they can respond to stimuli, use energy, adapt to their environment. Overall, as a species, they will change somehow over time. Make sure that you also know what all of those vocab words mean that I just mentioned that were part of any of those characteristics. Now, we talked about the metric system, why we use the metric system instead of the standard system that the United States uses. One, it's so everybody can be on the same page and keeps it universal so we can share data with people across the world and they can understand it, even if they can't understand all of our words sometimes. But 2, we said that converting metrics was super easy because of the powers of 10. And you remember that silly little phrase, King Henry died by drinking chocolate milk. And then remember, I added 3 more places to the right for a special unit. And all of these, remember, are the prefixes, except for the B. The B, remember, is the base unit in the metric system. Whether you're talking about meter for distance or length, liter for volume of a liquid, usually, and gram, which is the mass of something. So those being the base units, you can put any of those prefixes that you want. Now, remember there's 2 D's. So I always remember that the one on the left is DA. Remember that one's deca. And I kind of remember that one because there's an A. Like A comes before the other one. Okay, so try a practice problem. I had one on the sway for you. We'll say, for example, 2.1 liters equals how many milliliters. Remember that you have to find where did you start. In this case, starting with L, no prefix in front of it, which means you started at the base unit, and you want to go to M, which is the milli. So you're going to go 1, 2, 3 places to the right, which means you're going to do the same thing with your decimal point here. So you end up with 2,100 milliliters. That's what goes there. And now remember, this is milli, but then this funny one down here, which is 3 places to the right of milli, is micro. That's that funny Greek letter mu that we discussed. Okay. Make sure you can do that without looking at your notes, you know, just remember that conversion ladder. Now, ecology. You've been spending quite a bit of time in class and at home the last few weeks studying ecology, both with me and since I've been gone. Make sure that you're studying all of the ecology assignments I gave you. Go back and re-watch the video clips from YouTube that I posted on Edmodo for your group, for your class. Those are the videos that you watched actually during some of your class periods. They're only like 5 or 10 minutes, so they'll do a really great job of helping you refresh your memory as you're preparing for the test this week. I feel like most of you are pretty confident on the basic ecology stuff. Autotroph versus heterotroph. I'll do a silly little example to refresh your memory though. I'm going to draw a food chain. Very simple one, like one you've seen before. You should be able to, be able to create a food chain and also know how to read them. Obviously, the carrot, that's a plant, so it's an autotroph producer. The thing that's eating the carrot, remember we said, is a primary heterotroph, aka primary consumer, the rabbit. The thing eating the rabbit, the snake, is a secondary heterotroph, consumer. Remember, they mean the same thing. And then the hawk that's eating the snake being a tertiary. Can there be something above the hawk? Yes. There's possible to have a fourth level, a quaternary structure. We didn't discuss it in class. It's not commonly talked about. But you can have another tier to your pyramid and to your food chain. Now, if you were to take all of the food chains in an ecosystem and put them together and have them overlap each other looking like cobwebs, remember that's a food web showing you all of the interconnected feeding relationships. And now remember, these things, autotroph, heterotroph, these are the trophic, remember trough feeding. These are the trophic levels of the ecosystem. And so when you guys made that foldable, that awesome pyramid, I'm not going to spend a lot of time on this because you spent a lot of time in class on this. Whether you want to call this an energy pyramid, a pyramid of numbers to tell you about the population size, or a mass pyramid to show you about the mass of these organisms in the ecosystem, it's all the same thing. The pyramid in a balanced ecosystem should look kind of like this, with down here being your autotrophs or producers. They should be the highest concentration. And remember, they're getting their energy directly, in most cases, from the sun. Now, I'm going to show in black the energy transfer. Just like these arrows up here in the food chain are showing that when the rabbit eats the carrot, the energy is being transferred to the rabbit. Same thing I'm going to show down here. We're going to call this an energy pyramid. So the autotrophs, the plants, are getting their energy straight from the sun. They're using it to create food. And then whoever's eating them, the primary heterotrophs, when they eat them, it transfers the energy. And remember, we said that only 10% of the energy gets transferred to the next level. What happens to the other 90%? Utilized in metabolism and lost to the environment as heat. Okay, so next, when the secondary consumers eat the primary ones, their energy is getting transferred to that level, and so on to the tertiary. And you could draw a fourth level and do the quaternary. Remember, only 10% gets transferred. If you were to take in any ecosystem, whether it's energy or numbers or mass, and flip this pyramid upside down, so that instead the little part was on the bottom and the big part's on the top, I want you to think about for a second what that means. So if now you go from the autotrophs, the green stuff, being in highest numbers, to now the autotroph being in the lowest numbers, hopefully you're starting to think, hey, there's something wrong with that. We already know if I take a triangle and try to put it upside down, it's going to topple over. This pyramid's going to fall over. There's nothing for it to stand on. It's too skinny. And the same thing happens in the ecosystem. If something happens to mess with these autotrophs, these plants, and starts wiping them out, where there's less of them, your ecosystem's going to start to collapse. Sometimes very quickly, sometimes slowly, just depends on the organisms that live there. So you want to have balance, and you need to have the autotrophs being in highest number in that ecosystem. And whoever is these high-level predators that are up here in the third level, they should be the smallest quantity of anything, because it takes an awful lot of organisms to give them the energy that they need to be able to survive. Now, we didn't talk about this much together, so I'm going to tell you about symbiotic relationships now. Basically, that just means organisms are living in close association with each other. And for symbiotic relationships, there's three basic ones: mutualism, commensalism, and parasitism. Refer to your review sheet so you can follow along with me. So if they're having a symbiotic relationship with each other, they're very closely related to each other. They have a very close living relationship. They need each other. They rely on each other heavily for something. In mutualism, it's like I tell you, if you and I are talking about something and we say that the feeling is mutual, then this is a plus-plus relationship. Both parties benefit. We both feel the same way. We're both in accord. We're both happy about it. This would be like the clownfish and the anemone. They both get something good out of the deal. A parasite, like a tick on a dog, obviously the tick's getting food and a nice warm place to live. He's happy. The dog is ultimately getting harmed, minus, so, because obviously getting your blood sucked, okay? Now, does a parasite kill its host? It doesn't want to, okay? Because if you think about this, if the parasite kills its host, then it loses its meal ticket and its place to live. Parasites can kill their host, although that's not their immediate goal. Commensalism, one of them benefits and the other one gets nothing out of the deal. So the nothing out of this would be like barnacles living on a whale. The whale gets absolutely nothing out of the deal. The barnacles don't hurt it, but yet the barnacle is getting, well, it's kind of protection because it's living on one of the largest creatures in the ocean, but also it's being exposed to large amounts of territory, and it's getting a more diverse like food source available to it. So that one is a +0. Somebody benefits, somebody gets nothing out of the deal. And then there's another kind of relationship. It's not symbiotic, but it's called predation, and that's the one they taught you about in elementary school. Everybody knows predator-prey relationships. That one's also a plus-minus, right? The snake's happy about the fact that he ate a rabbit. He got food out of the deal. The rabbit got harmed so much that he got killed. So when you look at predation being plus-minus and parasitism plus-minus, you go, well, they're kind of similar. And they are kind of similar, except for if you're a predator, the end goal is you actually kill your prey and you eat them. That's the difference. And they don't actually live with each other, where a parasite would live with its host. Remember the hierarchy of the levels of organization of life. I'm not going to write it out just due to time now. We start out with the individual organism, and a group of organisms that are the same species living in a given area make up a population. Similar populations in an area make up a community. Similar communities living in the same area make up an ecosystem. Now, an ecosystem has all the communities, all of the different living populations, but the ecosystem also has something else. Okay, so there's two things in the ecosystem: the biotic, the living things, and the abiotic, the non-living things. We talked about this before. A means not. So the ecosystem has all of the living communities, all the biotic factors, all the organisms present, but there's also the abiotic factors like wind, water, temperature, etc. I'm not going to talk much about the carbon and water cycle. You guys should have spent quite a bit of time reading, color coding, and looking up terms that are associated with them. But there's a few buzzwords that I want to make sure that you understand. We'll do carbon cycle first because it's probably the one you know the least. A few terms like respiration, combustion, photosynthesis, so you know that right now what we're doing is respiration. We're breathing. Oxygen in, CO2 out. Okay. So on your drawings, you have pictures going from animals up to the atmosphere, up in the air, showing that you're releasing CO2, carbon dioxide. Now, if you look at your pictures, you're going to see arrows going from the trees up, releasing CO2, which might seem a little strange to you because most of you are very much aware already, prior to you ever setting foot in my classroom, that plants do photosynthesis because they're green, right? Which you know that means that they take in CO2, the stuff we're spitting out, and they release oxygen. Okay? I would make the arrow go up in the air, but it's just confusing on my picture here. Doesn't matter. So now, that's the basic premise behind photosynthesis. But what they probably didn't teach you back in elementary and middle school about trees and other autotrophs is that they're doing this photosynthesis to make food. You know that part. They're breathing in CO2, breathing out oxygen as a waste, and then we get to breathe it, which makes us live. But they also do respiration. So that respiration business is not just for animals like us and other heterotrophs. It's also for autotrophs like plants. Because once they make their food, they still have to be able to take that food and turn it into a usable form of energy, just like you and I. We'll learn more about that in a few weeks. So what's going to happen is they also are breathing in oxygen, breathing out CO2 through respiration, just like you and I are right now. And if that seems strange to you, because nobody ever told you that a tree spits out CO2 just like us, it's because they release more oxygen than CO2. Combustion means you're burning something. Whether you are having a bonfire in your backyard, driving your car and we're doing combustion of fossil fuels, or a factory, or whatever, electricity, whatever needs to happen. Those things that are actually burning fossil fuels or carbon in some way, burning wood, that's burning carbon, right? Because all living things on our planet have carbon in them. This is releasing carbon up into the atmosphere. And while I'm on this, then I will tell you that CO2 being released up into the atmosphere kind of makes like a little blanket in the upper atmosphere. And it's like putting on a comforter at home. The thicker your blanket, or the more blankets you put on, you get warmer and warmer. Well, the same kind of idea happens with our planet, along with like in an actual greenhouse where somebody wants to grow plants, like especially if they live up north and it's too cold, and they make this special room, and it's, you know, usually glass or something like that to keep the plants from getting too cold. And the idea with this greenhouse is the same thing that happens with the CO2 being released into the atmosphere by us and other organisms on the planet, is we're releasing the CO2, which makes a little blanket up in the upper atmosphere, and it makes us kind of warm. More CO2, thicker the blanket is. That's a good thing. It keeps us from freezing to death. But the problem is, on our planet, if you create too much of that, then you can cause some climate change by having too thick of a blanket on, just like you can start sweating if you have too many comforters on. So that same premise that keeps plants in a greenhouse nice and warm up north keeps these guys, or keeps our Earth, I guess, the right temperature for us, but can also make it too hot maybe at some point. That's to debate another day, not today. On the water cycle, I'm going to say there's probably one term that was brand new to you. And on the water cycle, that term is transpiration. That's really a fancy word that basically means the plant is sweating. Not because it's hot, but it's releasing water into the atmosphere. You've seen dew collect on a outside of a leaf or something like that. Anyhow, that's for another day. I do want to real quickly tell you about population growth, but as we're getting ready to wrap up here. So there's two kinds of population growth. This one, graph A, which is that J-shaped curve where the population is just doubling, keeps increasing. That's exponential growth. Over here, though, on graph B, is more, it starts out looking like a J, but then it kind of levels off like a flat line up there, showing that the population is not getting bigger, it's just staying the same at that point. So these two graphs are showing you the two major types of population growth curves that we study in population ecology. Graph A being exponential growth. Nothing's stopping it. It just keeps doubling. In real life, that doesn't happen so much, except for like maybe in a petri dish with bacteria, although the human population is starting to look like this right now. Meaning there are no restricting factors, nothing that is going to stop the population from growing out of control. Over here, something happens where you end up with it leveling off. And that's real life, what generally happens in an ecosystem of any type. And this is the one that's called, in graph B, it's called logistic growth. And so this dotted line that I've drawn, where you see a flat line, it doesn't mean that the population is zero, because zero is down here. It's not zero. It just means it's not growing. It's staying the same. And remember, that point where it stays the same is called the carrying capacity. That's the max number of individuals that can be supported by that ecosystem. Why does that happen? Why is there a cap? Well, we have limiting factors. Whether they are density dependent or density independent, there are still limiting factors that restrict a population's growth. That's real life. The size of the room in my classroom restricts how many people can be in there. We can't fit 100 people in there. You probably wouldn't be able to breathe real well. Temperature, amount of food available, amount of water available. How many predators are around. There's so many things that can be considered limiting factors. Anything that kind of helps keep the population numbers in check. Diseases even can be limiting factors. So I mentioned a term that you and I haven't spoken with in person about, but those are the density dependent and density independent terms. And I just want you to think about for just 1 second as we're wrapping up here. Density. And I don't mean density like they taught you about probably in a physical science class. Not exactly, anyway. But similar idea. When we talk about the density of a population, we're saying, how many people are there in this one given unit of space? So if I have, let's see, this side of my classroom with all of my desks in it, and let's just say there's 25 of you sitting there, okay? And then I have over here my lab benches where the windows are, and let's just say there's only one person over there, because you know I like to stand over there sometimes when I'm talking to you. So you see there's a human population on both sides of my classroom, but this side has more people in pretty much the same amount of space that's on the other side. So this side is much more dense than that as far as human population goes. So now, what's something that could restrict the size of this population over here? Say where you're sitting in my classroom, okay? If we were locked in there and we couldn't leave for a week, amount of food available, that could be a problem, right? Amount of water, maybe not a huge issue because I have sinks as long as the water's working. Are those things, amount of food and amount of water, and are those density dependent or independent? Is it the same effect if you're here or if you're here? So, if I bought one pizza for the whole class to share, and I bought one pizza for me over here hanging out with my turtles to share, with just myself, because I can't give them turtles. They're not Teenage Mutant Ninja Turtles, right? Okay, not going to work. So, is amount of food, the pizza, the amount of food available to those people on that side of the room, is that dependent on how many of them are there? Oh yeah, right? Because this is me, I get the whole pizza to myself. Yum. You guys are going to fight over these 6 pieces of pizza, right? And your population size, if we were left to stay there for a long time, would start to dwindle down because there's not enough food available. Density independent, things that are not affected by the population size, they might be things like, I don't know, maybe the temperature of the room, although that could be affected by the population size too. Think of something like if a flood came by. Is the flood going to devastate this side of the room and this side of the room equally? Yeah. The flood doesn't care if there's one person there or 25 or 2,500 people. The flood's still going to decimate the area. So things that are natural disasters are most definitely density independent. They don't care what the population size are. They're still going to have the same exact effect. Okay. I'm not going to get into the human impact on the biosphere because you're smart people and you've been studying this your entire life. Go back and read a few things in your book about it. But I will say this. I put a term in there called sustainable development. I want you to pay close attention to that term. Because sustainable development is when you and I as humans are practicing smart, earth-friendly practices that help protect the planet, not just for us, but for the people to come behind us. So I'm practicing sustainable development if I shut off the lights in my house in the room where I'm not hanging out. If I take a shorter shower. If I unplug my phone charger when I'm not using it. That's sustainable development. If I am making sure that if I have to bulldoze a part of the forest or the trees on my property, that I go and replant trees somewhere else nearby, so that I'm helping to replenish or conserve on the resources on the planet somehow. That is sustainable development. So take a look at that, and whether those resources I talked about are renewable versus non-renewable in our lifetimes, because gasoline and oil, those are not renewable resources. Although we can, over time, our earth can make more for us, it's not going to regenerate it in our lifetimes quick enough for us to have them. So we have to be very careful about how much we're using and how much is available to us if we want to continue to use those things. If you're using wind or water or solar power, that's sustainable development. Okay. Anything else you can think of that you want to talk about, message me on Edmodo, I'll get back with you. As far as biochemistry goes, I am not going to talk about that today because it doesn't count for your quarter grade in my class on the first part 1 test I discussed with you. It'll just be on that benchmark test, and you and I next week, hopefully, will be face-to-face and talking in detail and doing some fun labs with biochemistry so you can learn more about it. So, thanks, and hope you have a great day.